Hi kids, am I audible, visible? Am I audible, visible? Hello? Hi everyone, am I audible? Am I visible to you all? Good evening, good evening, good evening. So hi, welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma. I hope I'm clearly audible and visible to you all. Hi, good evening all. So today we are going to start a new chapter, that is structural organization in animals, right? Many students, they requested me for this particular chapter, so that is why I started it. And yes, many of you are asking for the plant kingdom part 2. Don't worry, I will take a live session on that. Okay, I will not be able to, I cannot upload the same video again, but yes, I will take the another session. Okay. Okay, I will take the another session. And why these days you all are after the plant kingdom, plant kingdom, plant kingdom? What about other chapters? Are you people done with that? Huh? Okay, so today let's start the chapter. And guys, here you people can see, right? You people can see the various videos that we are uploading for your information, for your motivation, for your concentration as well. So here you guys can see, bache, must watch these videos, and here you guys can see the video that I have uploaded for the tricks of the plant kingdom, right? So watch this video. It is just for 8 minutes and you will get all the tricks to remember the examples of the plant kingdom. And same I will post for the animal kingdom as well. And kids, let me tell you, entries for NEET Avengers batch, it is entries for NEET Avengers batch are already closed, you know that. And now we are soon going to close the entry for the NEET Energized batch as well. So 5th August, that is tomorrow, is the last day, right? And tomorrow is the last day to enroll there in that NEET Energized batch. So what you have to do, just go to this video, right? You can check any video, and then in the description box here you people can see the link for the NEET Energized batch as well, for the All India mock test series as well. So click on this link, add your number, add your email, and obviously you will get this batch at a price of 14999. Done? Okay. Chal. So now let's start the chapter, and it is the structural organization in animals. What is the chapter name? It is the structural organization in animals. Here, bache, I would like to tell you something. Please, first of all, please open up your NCERT. So it's structural organization in animals, right? I hope you people are aware that there is one more chapter, structural organization in plants as well. And in that structural organization in plants, we talk about the morphology and anatomy. So here in this particular chapter, you will talk about the tissue and you will talk about the cockroach. Okay. Okay, you will talk about the tissue and you will talk about the cockroach here. Clear, bache? Let me tell you. See, note down. Tissue, all the tissues. And then we will talk about the cockroach. So many times students, they ask us that, ma'am, what about the frog and earthworm? But a frog and earthworm is not the part of your syllabus. No doubt, last to last year, few questions were there. I think 1 to 2 questions were there from that frog or earthworm part. I don't remember exactly, but yes, questions were there. So this frog and earthworm part, it is also related to your animal kingdom. So sometimes, you know, in relation to that, they ask the question. But actually, if you check the information bulletin, right? If you check the information bulletin of last year, right? So as for that, it is the cockroach which is the part of your syllabus, right? And now, this part, cockroach, from this particular portion, you will hardly get one question. Okay, you are going to get only one question. So we will cover this portion near to the exams. Like in the month of January, February, March, I will take the class on the cockroach, right? One short type class, so that you can revise everything from this part. Okay, so my advice is focus here, right? Complete the tissue part because it is important for the human physiology, and might be we'll start human physiology next, and you should know about the structural organization part. Right? So this is what we need to study here in this particular chapter. Now, first of all, let's talk about the life. All right. So now when you talk about the living organisms, you know one thing, isn't it? You know that it is the cell which is the basic unit of life, isn't it? This is what we all know, that it is the cell which is the basic unit of life, isn't it, Pratibha? And this cell is going to, let me write it here so that you can revise it later. So it is the basic unit of life. It is the basic structural and functional unit of life. You can see any living organism, right? Below the level of cell, there is no independent existence of life. Okay, below the level of cell, there is no independent existence of life. So you can see unicellular organism, you can see multicellular organism, but basic unit of life is going to be the cell. So single celled organisms are also there, multicellular organisms are also there. This is what we already know. Now, when these cells, they join together, right? Like so many cells together, or you can say that a group of cells. I will discuss the conditions as well. So group of cells, what is it? Group of cells. Group of cells is going to form the tissue, the next part. The group of cell is going to form what? The group of cell is going to form the tissue. So if I have to define tissue, what will I say? Right? If I have to define tissue, what am I going to say here, bache? It is a group of cells. Just a minute, kids. Okay, sorry for this, bache. Yes. So when you talk about the tissue, tissue is what? Group of cells, right? It is group of cells. So these cells can be similar, they can be dissimilar. This is important. It is basically the group of cells. Cells can be similar, they can be dissimilar, right? Or you can say that group of cells having same function, having same origin, like this. Right, we will discuss this part in detail. So as of now, you just need to keep in your head that it is the group of cells. Then after it, what is going to happen, bache? So many tissues together, right? So many tissues together will form the organ, and so many organs together will form the organ system. What will they do? They will form the organ system, and then there will be an organism, a complete living body. Are you getting it? Then there will be an organism, a complete living body. So this is the hierarchy here. This is the biological hierarchy here, that basically it is the protoplasm having all the physical basis of life, right? It will form that cell part, right? It is forming that cell part. Okay. And you know that organic compounds and some inorganic compounds are making the structure. So cell, tissue, organ system, organism, that is what we have, isn't it? That is what we have. So if I relate the same thing, right, because right now we are talking about the structural organization in animals. So if we talk about these animals, okay, if we talk about these animals, so you know that even in animal kingdom, this is what we have studied, bache. Even in animal kingdom, you know that there are different levels of organization. There are cellular level of organization is there, tissue level is there, organ level is there, organ system level is there. Are you aware of it? Yes. Yes, bache. So when it is a cellular level, like your sponges, like your porifera, you know that here you will only see a multicellular body, right? Only you guys are going to see a multicellular body, and here you will not even see the tissue. You're not even going to see the tissue here. Then tissue level of organization is there, if you remember, Cnidaria and Ctenophora. Cnidaria and Ctenophora. Then organ level, platyhelminths. Okay, platyhelminths. And then from Esculminth to Chordata, yes, from Esculminth to Chordata, what do we have, kids? It's the organ system level of organization, isn't it? It is the organ system level of organization. So ultimately the cell, the tissue, they are making the complete structure. Isn't it? Isn't it? These tissues are going to form the organ system, and then there will be the body. So in this particular chapter, we are going to talk about the tissues and bache, study of tissues. is known as histology, right? Study of tissues is known as what? It is known as histology. What is it? It is known as histology. Okay, what is it, kids? It is known as histology. Is that clear? Any doubt here? Any doubt here? So this is a basic introduction, but all the points are from your NCERT only. Okay, all the points are from your NCERT only. So what are we going to start now? We are going to talk about the tissue now, right? What are we going to start with? We are going to start with the tissue, right? Done, bache? Done, bache? Okay, so this is what we need to study, the tissue. Okay, now it is also important, so let's relate here. So now when you say tissue, write down the definition. Study of tissue is histology, but when you say tissue, it's a group. It is a group of cells having similar structure, function and same origin, right? What is a tissue? Tissue is a group of cell having similar structure, function and same origin. Having similar structure, function and same origin, that is what a tissue is. Now this point is very important. Like see, in the case of tissue, students, okay, in the case of tissue, it's not mandatory that always the structure should be similar, right? In the case of tissue, structure can be similar or dissimilar. Okay, structure can be similar or it can be dissimilar. Fine, even function can also change. That's important. Function can also change, but origin should be same. Okay, origin should be same. Is it clear? Origin should be same. Is it clear? Right? So I'm repeating this again. It's a group of cells having similar structure, function and same origin. Right? Right. Structure can be similar or dissimilar. It doesn't matter if structure is even dissimilar in a case of tissue, but the origin of cells should always be same. Okay? The origin of cells should always be same. Done, bache? Done, bache? That is what a tissue is. Isn't it? Isn't it? Yes or no? Yes or no? So dissimilar structure and function is possible, but the origin should be similar. Origin should be similar. Done, bache? Yes? Clear? So obviously how these cells will connect to each other. Like let's say once, so as I said, group of cells it is. So in between these cells, what will you see? You will see intercellular substance, and it is also known as extracellular matrix. Okay, here what you people are going to see? You will see, yes, in the surrounding here, what you people will see? You will see intercellular substance or intercellular matrix. Okay, also known as extracellular matrix, which is rich in protein. Okay, clear? So within the cell, it's intracellular matrix. Intra mane within. Okay, and outside the cell, this is the intercellular substance, extracellular matrix, which will help in the attachment, which will help in the transport. So this is how you guys are going to get the tissue. Is that clear? This is how you people are going to get the tissue. So this is all about the tissue. Now, bache, it's very simple. You know that we all are evolved from us. We all are developed from a single cell, that is zygote, isn't it? We all are developed from a single cell, that is zygote. Okay, and this zygote is a totipotent cell. What type of cell it is? It is a totipotent cell, isn't it? It is a totipotent cell. Now, what is the meaning of being a totipotent cell? That it is having the ability to form a complete individual, isn't it? Now, this zygote, it will divide, it is going to form a multicellular structure. So, after many rounds of division, after cleavage, you know that you will get a ball-like structure, the morula, the blastula, the gastrula. I hope you guys are aware of it, isn't it? I hope you people are aware of it. Yes. So we will have the zygote. Zygote will divide. It is going to form a multicellular structure after the cleavage, of course. Right? We have completed that in human reproduction. You will see the blastomeres there. You will initially see the morula. Right? The blastula, morula, 8 to 16 cell stage, blastula, 16 to 32 cell stage, then 64 cell stage, blastocyst will be there, then gastrula will be there. So there are different stages of embryonic development, and after, so here during this embryonic development, you will see the gastrula. What will you see? You will see the gastrula. So, do you know gastrula? Do you know gastrula? Yes, kids? Right, so this gastrula will form. It is having what? Germ layers. The formation of germ layers is gastrulation. So during the embryonic development, we also see, okay, during the embryonic development, we also see the formation of germ layers, right? They are the extra embryonic layers. So they are 3 in number: ectoderm, mesoderm, endoderm. Clear? And these germ layers, they form different type of tissues. That's why I'm explaining it here. Clear, bache? That is the reason I'm explaining it here. Okay? Okay, so as I said, in the animal kingdom, different type of animals are there. Even we humans are also the animals. You know that the story will start from porifera. The story will start from sponges. The simplest type of animals having just cellular level of organization. Then comes the tissue, then organ, and the organ system is there. So here it is the structural organization of animals, right, bache? So here you know that you will see the organ systems, right? So you will see the triploblastic organisms here. So here in this chapter, mainly you are going to talk about the humans. Okay, you are going to talk about the humans here. Done, bache? So you know that we humans are what? We humans are triploblastic, right? We humans are triploblastic. 3 germ layers are there. So being triploblastic, we have ectoderm, outermost layer, mesoderm, middle layer, endoderm, innermost layer, and all of them will form the tissue. Clear, bache? All of them are going to form the tissue. They are going to form the different types of tissues. So I hope up to this part it is all clear. So let's revise it from this slide. So animals, as I said, they are composed of specialized cells which are organized into, right, which are organized into tissues that have different functions. Clear? Clear, bache? So animals, they consist the hierarchies of level of organization I just told you, and tissues are an integrated group of similar cells that perform a common function. So here in the NCERT also, this is what you people can mark, that cells, they can have dissimilar structure, they can have different function, but the origin should always be same, bache. The origin should always be same. So organs, they perform a specific task, right, and they contain 2 or more tissues. I will explain this part separately. So organ system mein, you know that it consists of multiple organs that together perform a vital body function. So this is what I have explained so far. So now let me give you the example of the organs. You know about the alimentary canal, isn't it? You all know about the alimentary canal, isn't it? Isn't it? Yes. So when we talk about the alimentary canal, when we discuss the alimentary canal, and when we talk about the histology, that is the tissue layers of alimentary canal, there is a trick here. Do you know the trick? It's SMSM. What is the trick here? It is SMSM. You know na, SMS, short messaging services. SMS these days, so we are using the WhatsApp, but SMS is also very useful for the OTP, for bank related OTPs, for delivery OTPs. है ना, SMS. So instead of SMS, you have to make it SMSM, right? What is it? It is SMSM. Okay. So S, outermost. This M, it is innermost. Now here, you will see serosa, which is also known as visceral peritoneum. Serosa, which is also known as visceral peritoneum. M stands for muscularis, means muscles are present there. Okay, S stands for submucosa, and even you will hear M stands for mucosa. Innermost layer, it is what? It is mucosa. So here, bache, this alimentary canal, and this part specifically the stomach, right? This part specifically the stomach, which is an organ. So here in this organ you will see the serosa, visceral peritoneum, muscles are also present, submucosa is also there, mucosa is also there. So here in these layers you will also see that nerve cells are innervating, isn't it? You will see that nerve cells are also innervating, like nerve supply is also there, right? We will also see the blood supply. So basically different types of tissues, they are making the organ. From this example, this is what you people can conclude, yes or no? From this example, that's what you guys can conclude, that organ is made up of different types of tissues. Okay, organ is made up of different types of tissues. Is that clear? Are you getting it? Yes, everyone. Are you getting it? So this is the meaning of this particular line. So we have different types of tissues, bache, right? So here on the basis of structure and function, okay, on the basis of structure and function, this is the word, on the basis of structure as well as function. So we have 4 types of tissues: epithelial tissue, connective tissue, muscular tissue, and the neural tissue is there. Clear bache? Clear bache? So we have epithelial tissue, muscular tissue, connective tissue, and the neural tissue. So this is what we need to study today. This is what we need to revise today. Clear bache? So so on the basis of what? On the basis of structure and function. So please take a note of it and just give me 1 minute, okay? Then I'll start. But tissue topic is not there in the new NCERT, but it is the part of old NCERT. Okay. So you really need to understand it. Fine. So on the basis of structure and function, we have 4 different types of tissue: the epithelial, the connective, the muscular, and the nervous. So bache, epithelial, epi means above, upon, right? It is present over something, like it is meant for the protection. What is the role of this epithelial tissue? We will see. It is meant for what? It is meant for the covering, for the protection. Isn't it? Connective tissue, obviously, to connect, to join. So it is connecting to different cells, right? Like blood is also a type of connective tissue, and you know its function. It is also playing role in the transportation as well. Now, muscular tissue, muscles, very important. They are for the movement as well. For that contraction and relaxation of muscles is responsible for passing the food to our system, to our stomach as well. Isn't it? Nervous tissue, very important for the coordination. It is the one which will send the message. Isn't it? It is the one which will send the message. So when you talk about the connective tissue, it will provide the support to the body. It is connecting the things, right? It is connecting to different cells. It provides support to body, isn't it? And I have given you the example of blood as well. So it is also playing role in the transportation, isn't it? It is also playing role in the transportation. Isn't it, bache? And when you talk about the muscular tissue, so it helps in what? It helps in the contraction and locomotion. It helps in the contraction and locomotion. right? And then comes what? Then comes the nervous tissue. So when you talk about the nervous tissue, so it is going to pass that information. It is going to conduct the information. So nervous tissue is to generate and conduct nerve impulse, right? So you know that all of them are very, very, very important for the proper functioning, for the proper coordination of the body. So this is about the tissue, right? This is about the role of the tissue. Now in detail we have to discuss it. Done, bache? Done? Clear? Okay, so please take a note of it as well. Done, bache? Done. So 4 types of tissues are there, and we have to start with the epithelial tissue. Clear, bache? We have to start with the epithelial tissue. Done? So epithelial tissue. What is the word here? It is the epithelial tissue, bache. So this is that tissue which originates first during embryonic development. Let me tell you. Epithelial, epi means upon, as I said. So obviously it is a tissue which is going to cover the body organs. It is meant for the covering, it is meant for the protection, bache. And here in this epithelial tissue you will see that all the 3 germ layers, right? Initially I told you about the germ layers, I told you about the extra embryonic membranes like your ectoderm, mesoderm, endoderm. So here in this particular tissue you will see that actually all the 3 germ layers are making this tissue. Your ectoderm is also making epithelial tissue, your mesoderm is also making epithelial tissue, your endoderm is also making epithelial tissue. Like I'll give you one example. When you talk about the mesoderm mainly forms your muscles, right? Your muscular tissue is mainly developed from the mesoderm. Few exceptions are there, but actually this is a scenario. But when you talk about the epithelial tissue, it is made up of all the 3 germ layers. Clear, bache? So what is the role of epithelial tissue? I want you people to answer quickly in the chat section. Yes, everyone, just answer it quickly in the chat section. What is the function of what is the function of epithelial tissue? It is meant for protection and the covering. Right, covering and protection, right? Which germ layer is forming the epithelial tissue? All the 3 germ layers are making the epithelial tissue, which it is the tissue which originates first during the embryonic development. So that is what you need to remember. So one by one we will discuss. Now first of all we will discuss the detail of this epithelial tissue, and then we will talk about the connective tissue, muscular tissue and the nervous tissue. So no doubt, bache, here in this part you will get the introduction only, and the detailed introduction of epithelial tissue will be provided to you. Okay, the detailed introduction of epithelial tissue will be provided to you, but other tissues like muscles, neural tissue and all, so I will tell you the basic structure, right? But we have to discuss all that things in the human physiology, so then your concepts will be more clear. Clear, bache? Then your concepts will be more clear. So let me write it here. So epithelial tissue, so tissue that originates first during embryonic development. It is the tissue that originates first during embryonic development. Clear, bache? Done. During embryonic development. Is that clear? That is what the epithelial tissue is. Clear, bache? Clear, bache? Yes. Yes. Now one line is given in NCERT that this tissue either faces body fluid or the outside environment, body fluid or the outside environment, thus it is what? It is acting like a lining, it is acting like a covering, yes or no? Yes or no? Yes or no? Right, this is the line from the NCERT now, that it is either facing the body fluid or the outside environment. So thus it is providing as a covering, isn't it? Isn't it? Right, so many times students are not able to understand the meaning of this particular line. So it's very simple. See, epithelial tissue, right? It is also lining your gut wall, right? Your alimentary canal, it is also lining your gut wall, right? Your alimentary canal, your digestive system wall. First point, it is also lining the skin here. What is it? What is it? It is the epithelial tissue. So obviously either it is facing any body fluid, it is facing what? It is facing any body fluid. Like, even let me give you one more example. Even in the blood capillaries, right? Blood capillaries are also covered with this epithelial tissue. Okay, they are also covered with this epithelial tissue, right? Right? So you will see that either they are facing the body fluids or they are facing the outside environment, right? Or they are facing the outside environment. Like if I talk about the body blood vessel also, so obviously internally it is also facing this blood, right? Externally, that extracellular matrix, that extra part is also there. So ultimately that is why on this basis we are saying that it is acting like a covering or like a lining. So any doubt here? Yes, everyone. Any doubt here? Tell me, do you have any doubt here? Yes. Any doubt here? So always remember, epithelial, epi means above, upon, thelia means growth. Okay, okay, so it will grow above other structures. Done, bache? Now when you talk about the epithelial tissue, you will see that here in this case, right, it is the only tissue where, you know, cells are arranged in the proper layers, where cells are so compact, they are arranged in the proper layers. You will see no intercellular spaces, or you will see very less intercellular spaces. Are you getting it? This is what you are going to see here in the epithelial tissue. Let me show you with the help of diagram. Like let's say this is the, right, just to make you this part clear, na, that is why I'm drawing it like this. So let's say, so as I said, in the epithelial tissue, what are we going to observe? We will observe the layers, proper layers, compact layers, right? You are not going to see such things in other tissues. First point. Here you will see the cells are compactly arranged with no or less intercellular spaces, and then bache, because we do not have the intercellular spaces here, so you will not see the blood supply to this epithelial tissue directly. Okay, you are not even going to see the blood supply to this epithelial tissue directly. So nerve supply can be there, some nerve cells can innervate, but blood supply is not there. So how this structure will grow, how will it get the nutrition? Now from this diagram everything will be clear to you. Okay, bache? So just below the layer of this epithelial tissue, right now, as I said, I'm teaching you the basic structure. What am I teaching as of now? The basic structure. Okay, the basic structure. Yeah, and yes, it should be clear. This is how it is present actually. Okay, like out. Epithelial tissue. Okay, so it is present just above. Okay, just a minute, bache. Yes, it is present. This is the epithelial tissue. It is present just above this layer. This layer is actually the basement layer. Okay, the basement layer or basement membrane. And let me tell you, this is acellular. Like it is not made up of any cell. It is not made up of any cell. It is made up of your glycoprotein, your collagen, your mucopolysaccharides. I will explain it. And just below this basement membrane, the part here is the connective tissue. What is it? It is the connective tissue, having abundant blood supply, having helping in the attachment as well. Now understand this part. Okay, I will write all the points, but first of all understand it. So epithelial tissue, epithelial layer, right? So no doubt you can see the cells are quite compact, no blood supply is there directly. So as I said, always this epithelial tissue, it lies on the basement membrane. Okay, it lies on the basement membrane. Clear, bache? Okay, basement membrane is there. And now when you talk about this basement membrane, it is having two parts. Like this part, this layer is the basal lamina. This layer is the basal lamina, right? I think it's not clear. Let me, so this layer, this which is in white color, it is the basal lamina. Okay, and just below this, right, here you have fibrous lamina. So basement membrane is having two parts. One is your basal lamina, another is your fibrous lamina. So I'm writing it here. Okay, one is what? Is your basal lamina, which is near to epithelial tissue. Another one is your fibrous lamina, which is near to the connective tissue. Now, as I said, this is acellular. So, bache, the basal lamina, it is secreted by your epithelial tissue and made up of glycoproteins. So it is secreted by epithelial tissue and made up of glycoproteins. So obviously no cell is present there. So 110% it is acellular. Now the fibrous lamina is near to connective tissue. It is facing the connective tissue. So it is secreted by what? It is secreted by connective tissue. So this layer contains, okay, it contains collagen and some mucopolysaccharides. Okay, some mucopolysaccharides. Are you getting it? So it is facing the connective tissue. Clear, bache? It is facing the connective tissue. So both of them together, they are making the basement membrane. Clear? Both of them, they are making what? They are making the basement membrane, and this is the basement. On this basement membrane, your epithelial tissue is lying. Clear, bache? Your epithelial tissue is lying on this basement membrane. Is that clear? Yes. Is it clear? Sure. Now I have used the one word here, that here you will see collagen. What is collagen? Can you tell me? I hope you guys have attended my biomolecule classes. What is collagen? But you always remember, collagen is a triple helical protein. It is the most abundant protein of animal world. What is it? It is the most abundant protein of animal world. Clear, bache? I'm repeating this again. Most abundant protein of animal world it is. And even you can say that it is most abundant protein of extracellular matrix. It is present in extracellular matrix. And this collagen protein is a triple helix. Okay, triple helical structure. And let me tell you, bache, your amino acid proline, hydroxyproline, and one more amino acid is there, which is and that amino acids are making this collagen. Okay, and when you are using the word mucopolysaccharide, so I hope you remember mucopolysaccharide, right? It's mucopolysaccharide. So in the case of mucopolysaccharides, heteropolysaccharides they are, right? Heteropolysaccharides they are. Clear, bache? Clear, bache? So here it is hyaluronic acid. Okay, it is hyaluronic acid, and this hyaluronic acid is a cementing material. So in the living body, in the living system, this is what you guys are going to find. So this hyaluronic acid, the cementing material is going to, right, it is going to join everything. Clear bache? Clear bache? Done? Yes, is it clear? So mucopolysaccharide is hyaluronic acid, the cementing material. We have studied it earlier. Remember when we talk about the egg membranes? In the case of egg membranes, we used to say the corona radiata, they are joined by the cementing material. Like in sperm content, in acrosome, we used to say na hyaluronidase. Hyaluronidase. So that enzyme used to break this hyaluronic acid, the cementing layer, so that sperm can penetrate. So we have discussed it there. Okay, we have discussed it there. Is that clear? Is that clear? Okay, done. So this is what you need to remember. So yes, this basement membrane is secreted by both. It is secreted by both your epithelial tissue as well as your connective tissue. Clear, bache? Clear, bache? Sure? Sure. So please mark this line that epithelial tissue is the only tissue, it is the only tissue having uniform layers, okay, having uniform layers and cells are compactly arranged. Cells are compactly arranged and little or no intercellular matrix is there, and even not even the spaces are there. Clear, bache? Clear, bache? So this is what you are going to find. So this epithelial tissue layer, it is going to take the nourishment from this connective tissue. Okay, so you know that the diffusion will occur here, right? So the essential things will diffuse from the higher concentration to the lower. So with the help of this from this basement membrane, right, connective tissue is having everything useful. So obviously from this basement membrane, all of them will pass to the epithelial tissue. Is that clear? Is that clear? Yes. Yes, bacho, is it clear? Okay, so cells are arranged in a uniform layer, and here cells are compactly arranged, so no intercellular space and no intercellular matrix is present here, right? So this point is very important, please note down. Done, bache? This point is very important, please note down. Fine? Sure? So here you guys can see, just look at it. So free surface, the free surface here, it faces the body fluid or the outside environment, so it covers or line the body or body parts. So cells are compactly packed with little and no intercellular matrix. So important MCQ it is. Fine, bache, it is very important. Done. So now the question is how these cells will, you know, then when they do not have the intercellular spaces, okay, that's fine, but they do not have the intercellular matrix. So how will they join? How will they join? So in the case of these cells, what will you see? We will see interdigitations, right? What are we going to see, bache? We are going to see the interdigitations, isn't it? Isn't it? Right? So you will see that sometimes the plasma membrane, right? Sometimes you will see the plasma membrane, it enters the cytoplasm of another cell. But that is majorly in the case of transitional epithelium. Here mainly we are going to talk about the cellular junctions. What are we going to discuss here? We are going to talk about the cell-to-cell junction. Okay, cell-to-cell junction. Done, bache? Isn't it? Isn't it? So the very first thing that you need to know is your, yes, it is your, can you tell me the name of these junctions? Do you know anything related to these junctions? Yes, anything related to these junctions? As I'm saying, the cell-to-cell junction is there, right? Cell-to-cell junction is there. So anyone? So basically, see, the cells are present. Like, let's say you have one cell, you have another cell. So how are they connected? But sometimes you will see some tight junctions here. Okay, sometimes you are going to see some tight junctions here, right, by which they are connected. Sometimes we will see, like, these are the types of junctions that we need to study here. So tight junctions we are going to talk about, right? We will even talk about your gap junctions, which are the continuous junctions, bache, right? We will even talk about the desmosomes. Okay, we are even going to discuss about the desmosomes. Fine. Fine. So tight junctions, gap junctions, desmosomes, this is what we need to study. Clear, bache? This is what we need to study. Done? Done? Yes. So जैसे let's start with the tight junction. As the name is clear, बच्चे. Tight junction. So tightly it is going to hold two cells together. Tightly it is going to hold two cells together so that there should be no leakage of substance. Okay. So tight junctions majorly you will see in the case of columnar cells. Where will you see it? You will see it in the case of columnar cells. Now the question is, ma'am, what type of cells are columnar cells? Bache, they are tall and cylinder, and the nucleus is present at here near to the base. Okay, these cells are tall and cylinder, and the nucleus is present near to the base. So in such cells, and especially in intestine, you will see, like let's say this is the one cell, this is the another cell. So here in this case, what is going to happen? See, the plasma membrane, it will fuse in such a way that you will see they are tightly fused. Okay, you are going to see the tight junctions here. So here it is tightly fused so that there should be no leakage of substance. Okay, there should be no leakage of substance. Is it clear? There should be, I'm repeating this again, no leakage of substance. Okay, okay, so in the diagram you see it like this, something like this. Right, that plasma membranes, right, plasma membranes, they are fused here. Okay, okay, so there will be no leakage of the substance. Now, if you notice, right, here, like in some cells, what is going to happen, that in the apical portion, right, apical portion will have tight junctions so that there will be no leakage. Apical part will contain tight junctions so that there should be no leakage of the substance. And here, this basal part is not having these type of junctions here. So sometimes some things can diffuse. So sometimes the junctions, they are present in this way that they can make these cells, right, they can make their both the surface both the parts different. Apical part means this tip region where tight junction is there so that nothing can diffuse from this part to another. And when you talk about the basal part, because here we don't see the tight junction, so obviously things can pass from this to this, right? So the same cell, it can behave in a different way when it comes to the transport of the things or the diffusion. So mainly what you need to remember, that tight junctions, they will avoid the leakage of the substance. Clear, bache? Clear, bache? Now the next is the gap junction. Gap junctions are, bache, continuous cytoplasmic connection. What are they? They are continuous cytoplasmic connection, right? You know, na, cells, they also exchange the substances. They also exchange the substances, isn't it? So when it is a gap junction, it is nothing, it is the continuous cytoplasmic connection. What is it? It is the continuous cytoplasmic connection by which the things will move. Clear, bache? So here in gap junction, right? Here in this gap junction, here, this junction, it is also made up of, this junction is also made up of protein, like it is connexin protein. Which protein is there? It is connexin protein. This is extra, bache, right? Which protein is there? It is the connexin protein, right? Connexin protein. So basically six connexin proteins are making this gap junction, this continuous cytoplasmic junction. So obviously things can easily cross here and there. So this tight junction, it avoids the leakage. It is acting like a cementing junction. It is joining two cells but avoid the leakage. The gap junction, the continuous cytoplasmic junction by which the things will get exchanged. So in the animal cell, okay, in the animal cell, this is what you people are going to see, gap junctions. Okay, gap junctions are just, you are confusing two different things. Okay, but in the case of plants, if you remember, we only talk about one junction. There is no, in the plants, you will not see tight junction, you will not see another junctions, you will only see one junction, and that is, right, and that junction is your plasmodesmata. Okay, which junction is there? That is plasmodesmata. Again, it is also a continuous cytoplasmic connection. Fine. Again, it is also a continuous cytoplasmic connection. Done, bache? Done, bache? So gap junction is there in the case of, it is there in the case of animals. Are you getting it? It is there in the case of animals. Done? Okay. Okay. Sure? Sure, sure. Any doubt? Yes. And the next is what? Next is the desmosomes. Next is what? Next is the desmosomes. Okay. Okay. So desmosomes, which are also known as adherence junction. Okay. So now in the case of desmosomes, basically desmosomes na bache, they help in the attachment of two cells, right? In the case of desmosomes, also known as, they are meant for adherence, obviously for the joining, for the connection, for the cementing. Okay, so it is also known as macula. It is also the part of these junctions. Macula adherence. Adhere means to join. So here in the case of desmosomes, what you people are going to see, like this is the one cell. Here you will see this, you know, this type of structure, this type of structure. Like obviously they all are proteinaceous, right? They all are proteinaceous. And here you will see some other, you know, protein fibers as well. Actually, in the case of desmosomes, right? In the case of desmosomes, what you have to keep in your head, that they are meant for the adherence, they are meant for the joining. Right? They are meant for the adherence, they are meant for the joining. So in the case of desmosomes, we see disc-like proteins. What are we going to see here, bache? We are going to see the disc-like proteins. Yes. What will we see? We will see disc-like proteins. Okay, disc-like proteins, and these disc-like proteins are also having these fibers, right? They are intermediate filaments. What are they? They are intermediate filaments. Okay, I'm repeating this again. What are they? They are intermediate filaments. If you have attended the cell chapter, right? I need to take the cell chapter yet. Yes, cytoskeletal element. We talk about the microtubules. We talk about the microfilaments. We talk about the intermediate filaments. So these intermediate filaments are basically what? They are the cytoskeletal element. Intermediate filaments, right, bache? So when you talk about the desmosomes, right, this is the example of a desmosome. So these desmosomes are having disc-like proteins. To these desmosomes, your intermediate filaments are also there. You will see keratin protein filaments here, and right, so they are meant for what? They are meant for the attachment of two cells. They are meant for the attachment of two cells. So this is what you need to remember. Which cytoskeletal element is there? It is the intermediate filaments, right? Which cytoskeletal element is there? It is the intermediate filament. Is that clear? Right? So we even have hemidesmosomes. One word is also there, that is hemidesmosomes. What do we have? We have hemidesmosomes. So when it comes to the hemidesmosomes, in their case, okay, in their case, what will you see? That hemidesmosomes, they help the cell to get attached to extracellular matrix. Right? Help the cell to get attached to extracellular matrix. Isn't it? Extracellular matrix. So it will help that cell to get attached to the surrounding. So obviously protein filaments are present there. Clear, bache? So desmosomes mein two cells are connected, disc-like protein intermediate filaments are there, and hemidesmosomes mein they will help the cell to get attached to the surrounding, that is to the extracellular matrix. Is that clear? Yes, bacho. Is it clear? So it is meant for joining, isn't it? Isn't it? Clear, bache? So these desmosomes, adhering junctions, they are meant for the cementing, to keep the neighboring cells together. Okay, to keep the neighboring cells together. Yes, done. Right, to keep the neighboring cells together. Done? Sure? Are you sure? Yes? Pakka? Okay. Right? So whenever you will see the multi-layered epithelium, right? We are going to talk about it. So especially in the multi-layered epithelium, when your epithelium is I told you na, epithelium tissue, epithelial tissue is the only tissue which is having proper layers. So when you will see the multi-layered epithelial tissue or the stratified epithelial tissue, so you will see that these desmosomes are meant for they are helping in the attachment, they are helping in the cementing. Okay, they are helping in the cementing, they are helping in the attachment. Is it clear? Yes, bache, is it clear? So this part is very important. Please mark it. Okay, this part is very important. Done. So now, bache, as I said that just a minute. Okay, so now as I said that this epithelial tissue is made up of, okay, this epithelial tissue is made up of three different germ layers, right? It is made up of all the three germ layers. Like it is made up of ectoderm, it is made up of mesoderm, right? It is made up of endoderm, isn't it? Ectoderm, mesoderm and endoderm. So when it is made up of ectoderm, right, if ectoderm is the germ layer which is making the epithelial tissue, then you will use the word epidermis. You will call it as epidermis. Okay, epidermis, you know na, epidermis, the outer covering. Yes, bache, it is the epidermis, the outer covering. Okay, okay. It is the epidermis which is the outer covering. Now, if mesoderm is forming the epithelial tissue, then you will call it as mesothelium. What is it? It is the mesothelium, right? It is the mesothelium. So it is going to form what? Right, mesothelium. Somewhere also related to the muscles as well. We'll tell you about it. Right, so it is what? It is the mesothelium. Right, bache? It is the mesothelium. Clear? Clear? So if it is the mesoderm which is making the epithelial tissue, so you will call it as, yes bache, please type it quickly, you will call it as mesothelium. Okay, you will call it as mesothelium. Done? Done? It is the mesothelium. Okay, and when it is the endoderm which is making it, then you will call it as endothelium. Okay, then what is it? It is the endothelium. Fine. Like just epidermis. Epidermis is mostly the outer covering. So you will see here it is going to be multi-layered, right? Stratified it will be. Stratified means multi-layered. It is going to be multi-layered. Now here, this mesoderm, it is made up of mainly this mesothelium is having simple squamous cells. Simple squamous cells, let me explain. Here also they are going to be simple squamous cells. Let me give you the example here. Okay, just say you know about the blood capillaries. Do you know about the blood capillaries? Yes. So whenever we talk about the blood capillaries, we always used to say that they are lined with the endothelium. I hope you remember. Right, whenever we talk about the blood capillaries, we used to say they are lined with the endothelium. So endothelium is nothing. It is also the epithelial tissue which is made up of endoderm. It is also the epithelial tissue which is made up of endoderm. And if I'm using the word simple, it simply means it is single layered. Okay, after this is what we are going to start. It simply means single layered. Okay, it simply means single layered, and you will see it is meant for the diffusion. It is meant for the exchange. It is meant for that exchange. It is meant for the diffusion. Clear, bache? Clear? Same as the function here in this mesoderm part, right? So mesothelium is also going to play a role like this. Done, bache? Done? Yes? Is it clear? Okay? Sure? Sure. So epithelial tissue is going to do what? It is going to do the lining, right? It will line, it will act like a covering. It will cover the body cavity, the ducts, right? Body cavity, the ducts. And moreover, bache, as I said, this mesothelium, you will see in the case of muscles, we'll relate it there. Okay, we will relate it there. So if there is any other doubt, please tell me. No doubt? Pakka? So can we start the next part? Can we start the next part? Yes? Sure? Bache, although one by one we will discuss, but still let me tell you one thing here, that these cells, these cells were having some outgrowths, some projections are also there. Like this is what we are going to study next. You will see that cells will be having some finger-like projections, and we are going to call it as microvilli. These are the different type of processes that we will see in the case of cells, right? Some cells, they will show what? They will show finger-like projections. What will they show? They will show finger-like projections. Okay, finger-like projections, microvilli. Okay, microvilli. So obviously it is meant for absorption of food. Done. It is meant for what? Absorption of food. In some cells, what are we going to see, bache? In some cells, you will see some, hair-like structure, something like this, right? The cilia will be there. Cilia. So cilia will move, back and forth. Cilia basically help in the movement, right? It will help in the movement. Clear, bache? It will help in the movement. Are you getting it? Cilia. So we will even see another process is stereocilia. Stereocilia is also there. That is a kind of tubular structure, not finger-like obviously, but tubular structure. Even we will see stereocilia. So this is nothing. These are the different processes that we will see on these cells, and this is what we need to discuss. Clear, bache? So microvilli, finger-like processes meant for the absorption, right? Your cilia is also contractile structure. It will help in the locomotion and movement. And your stereocilia, stereocilia is non-contractile, right? It increases the surface area. So as I said, you will see it like a conical structure. You will see it like a conical structure. So one by one we will talk about it. Done? One by one we will talk about it. So you will see different types of processes there on these cells. So if there is any doubt, do let me know, please. Yes. Done? Sure? Okay. Let's start the next part. Okay. Let's start the next part. And here you all can see this simple epithelium, single layered. This is what we need to discuss next. Take it, bache. So take out your pen and paper, and now we need to draw a flow chart. Okay. We need to draw a flow chart. Fine. Okay. Done. So let's start. Epithelial tissue. Okay. So bache, one is your simple epithelial tissue, right? It is going to be your simple epithelial tissue, and another is your compound epithelial tissue. Another is what? It is your compound epithelial tissue. These are the two words which you need to focus. One is the simple epithelium, right? Yeah, epithelial tissue. Another is the compound epithelial tissue or compound epithelium. You can even use word epithelium or epithelial tissue. So this compound is also known as stratified. Let me explain this. But a simple epithelium. Simple epithelium means it is single layered. What is it? It is a single layered. It is just made up of a single layer, isn't it? And when you are using the word compound epithelium, it is multi-layered. Okay, multi-layered, or you can also use the word multi-serrated. Serrated word is also used for the layers. right? You will also use the word serrated, multi-serrated. So single layered, also known as uniserrated. Okay, uniserrated, single layer is there. So this is how we are going to divide it. Simple epithelium or simple epithelial tissue, that means it is single layered, and another one is what? It is multi-layered. Done, bache? It is multi-layered. Clear? Clear? So now it is single layered, right? So obviously it will act like a covering which will help in the diffusion. Okay, so you can say that it lines, right, the body cavities. It lines body cavity, duct, etc. Okay, and here, because it is multi-layered, it is going to provide the protection. Fine, it is going to provide the protection. Done, bache? So now in the simple epithelial tissue, we need to classify it. Like you have simple squamous epithelium. What do you have? You have simple squamous epithelium. Okay, you have simple cuboidal epithelium. You have simple compound epithelium, sorry, columnar epithelium. Fine, and you have, it is one more category is pseudo stratified. It is pseudo stratified. Is it clear? It is pseudo stratified, right? So again, see, simple squamous, it depends upon, whenever the word is simple, it is saying it is single layered. Okay, it is single layered. Clear, bache? Clear, bache? Right? So next is simple cuboidal epithelium. Cube like cells are there. So it depends upon the cell shape. Okay? Okay? Then simple columnar epithelium. Then pseudo stratified. Fine, pseudo stratified. Okay, and when it is the compound epithelium, as I said, it is meant for protection. So here you will see two types. One is stratified epithelium, which will include your, stratified columnar and all. Another is transitional epithelium, which is also known as urothelium. Okay, it is the transitional epithelium, also known as urothelium. So this is what we need to study, and that is how we are going to divide this epithelial tissue. So now you know it is made up of all the germ layers, right, bache? And it is having this categorization. So we are going to start with this part, the simple epithelial. So simple means one thing is crystal clear, right? Simple mein one thing is crystal clear, and that thing is, it is going to be single layer. Fine, it is going to be single layer. Clear, bache? Clear, bache? Okay, done? Sure? Sure, bache? Any doubt? Yes, any doubt? So see, simple, composed of single layer, lines body cavity, ducts and tubes, composed of multi-layered, limited role in secretion and absorption. So mainly meant for protection. So we are going to start with the simple squamous. Okay. Simple squamous epithelium. But it depends upon the shape of the cell. When the cell is having a flat surface. Okay. When the cell is having a flat surface, right? It is having a flat surface. So like irregular boundaries are there. Some irregular boundaries are there. The surface is flat, right? Right. We also call this simple squamous. Let me tell you, we also call this as pavement epithelium. Okay, simple squamous epithelium. What will you see? You will see the cells with irregular boundaries. You will see the cells are having flat surface. You will see that cells, right? We also call it as the pavement membrane and the tessellated epithelium as well because of its shape. Are you getting it? Because of its shape. So what will you see? You will see a single layer of flat cells. Single layer of flat cells having irregular boundaries. Having what? Having irregular boundaries. Isn't it? Done, bache? Having irregular boundaries. So as I said, we will also use the another word, that is pavement epithelium or tessellated epithelium. Okay, you can also call it as pavement epithelium or tessellated epithelium because of its irregular shape. Fine, because of its irregular shape. Done, bache? Done, bache? So what will you see this? Where will you find it? You will find it wherever, you know, the maximum diffusion is there. Okay, you will see it wherever the maximum diffusion is there. Like, I hope you have seen the structure. I think even in 9th class, this topic is included. Structure is like this, right? Sometimes, you know, so you will see some polyhedral cells also. Done? Done? Okay, so it will be there. It forms the diffusion boundaries. Okay, it forms diffusion boundary. So wherever there will be the diffusion, you are going to see the simple squamous epithelium. जैसे best example is of your blood capillaries. Blood capillaries में you know that the exchange will be there. The exchange will be there. Your alveoli. Alveoli के case में I hope you know about it. You know na alveoli, the primary site for exchange of gases. Alveoli are the primary site for exchange, right? Primary site for the exchange of gases. What are they? They are the primary site for the exchange of gases in lungs. Fine. So in the case of alveoli also, you will see that they are lined with what? They are lined with the simple squamous epithelium. Okay. They are lined with the simple squamous epithelium. Fine. And here the blood capillaries, right, the which are innervating these alveoli, they are the endothelium. So now you know that when we use the word endothelium, it's just that this epithelial tissue is made up of endoderm. And in between these two, bache, because epithelial cells, they always lie on, right, the epithelial cells, they always lie on the basement membrane. So in between these two, you will see the basement membrane. Okay, you are going to see the basement membrane in between these two. Clear, bache? Clear, bache? So this is what you need to remember. So the examples are important. They can ask you it in the paper. Fine. They can ask you it in the paper. Done, bache? So this epithelium, as I said, mainly for the filtration and diffusion. So if you know in the body where these type of processes will occur, obviously you can name the examples, right? So in the alveoli of lungs, you are going to find it, right? Done, bache? Fine. Yes. Any doubt? Any doubt? Sure. So this is one example. We have another examples also. But do you know about nephron? Do you know about nephron present in kidneys? So in the nephron, do you know we talk about this Bowman's capsule? Do you know that? We talk about the Bowman's capsule, isn't it? See? See? This is afferent, if you remember, afferent arteriole. Okay. And this one is the efferent arteriole. Look at the diameter as well. So inner, see, this is the inner layer of the women's capsule. So, bache, here in inner layer of women's capsule, na, you're going to see some podocytes. Do you know that? When you talk about the nephrons, here in the case of nephrons, when you talk about the inner layer, right? When you talk about the inner layer of women's capsule, you will see, what will you see? You will see the podocytes. Okay, you are going to see the podocytes. Podocytes, poda word is associated with feet, right? So these cells are having some feet-like, you know, these cells are having some feet-like processes. Okay, okay, so here also, done bache. So here also in the lining of women's capsule, here in the lining of women's capsule, because exchange will be there, exchange will be there. So in the lining of women's capsule also, you will see simple squamous epithelium. Is that clear? Simple squamous, right? What will you see? You will see simple squamous epithelium. Is that clear? Yes. Done? Done? Alveoli, you know. I hope you know about it. Okay? And even bache, your body cavity. Body cavities, the spaces in the body, it is also lined with mesothelium. Now you know what is mesothelium. Fine. And you know about the blood capillaries also. So examples are important. Okay, Subhashini, what is important? Examples are important. Is it clear? Yes, sure. Yes, are you sure? Any doubt? Done, bache? Any doubt? So examples, again I'm repeating, they are important. And do you know inner layer of heart wall? In the case of your heart, the innermost layer is also made up of the simple squamous epithelium. Fine, it is also made up of simple squamous epithelium. Fine, so body cavity lined with mesothelium, you know, na, body cavity and even, you know, the body organs, that is visceral peritoneum and all, it is also made up of this simple squamous epithelium. So this is, that is how it is important. Clear, bache? So you can revise it quickly from the slides as well. So here you guys can see thin flat cells are present with irregular boundaries. So lines the walls of blood vessels and the lungs, right? So next is what? Next is the simple cuboidal epithelium. Next is what, bache? Next is the simple cuboidal epithelium. So as the name is suggesting, it's simple cuboidal epithelium. What is it? It is simple cuboidal epithelium. So cube-like cells are there, of course. Hai na? What will you see here? You will see cube-like cells. What will you see? You will see cube-like cells, right? Right. So cells are cube in shape, isn't it? Cells are cube in shape. Please note down. Yes, please note down. Cells are cube in shape. Clear, bache? Clear, bache? So it is meant for absorption, secretion and excretion. Okay, so when it comes to the function, so this epithelium is meant for absorption, secretion and excretion. Absorption, secretion and excretion. Clear? Clear, bache? Simple, right? What is it? It is simple cuboidal epithelium. Simple cuboidal epithelium. Clear? Sure? Sure? So as I said, examples are important. Okay, examples are important. So the best example that you need to remember is the tubular part of nephron. It is the tubular part of nephron. Fine. And moreover, in the thyroid gland, right, bache? even in the thyroid gland, even in the pancreas, you will see it. Okay? So your sweat glands, because see, the word is secretion, excretion, and absorption. So in sweat glands, you will see such cells. In the SNI of pancreas, SNI of pancreas means exocrine part of pancreas. This is the exocrine part of pancreas. You will see such cells in the exocrine part of pancreas as well, right? Right. So these are the examples. Now here, bache. Yes. One more thing. In the case of seminiferous tubule and in the case of ovary, if you remember, even in the case of ovary, even in the case of testes, we used to say na that they are lined with the cuboidal epithelium. है ना, that they are lined with the cuboidal epithelium. जैसे in the case of testes, right? In the case of testes, if you remember, right, when we take the inner section, just a minute. जैसे let's say this is the inner part. So here you know that in these testicular lobules, bache, in the testicular lobules, 1 to 3 highly coiled seminiferous tubules are present. Okay, here 1 to 3 highly coiled seminiferous tubules are present. So when we take the cross section there, this is what you guys are going to see, right? You will see the seminiferous tubule. And if you remember, in the testis though, we know that outer covering, here you will see the outer layer as, in the case of seminiferous tubule, you will see that cuboidal epithelium is lining it. And bache, this cuboidal epithelium, okay, this cuboidal epithelium here is known as the germinal epithelium. What is it? It is the germinal epithelium. Are you getting it? So it is a previous year question. Okay, it is a previous year question. Done? Germinal epithelium is what? It is the cuboidal epithelium. Cube-like cells are present. Fine. Cube-like cells are present. Clear, bache? Sure? Sure. Done? So in the ovaries also you will see this, right? You will see this cuboidal epithelium, germinal epithelium in the seminiferous tubule also. So why do we call it as a germinal epithelium? Because it is germinal, to germinate, to form something. Germinal, to germinate, to form something, right? Because it is going to form the germ cells, that spermatogonia and all, right? It will form that spermatogonia. In the case of testis, that spermatogonia and all. So that is why you are using the word germinal epithelium. Okay, right? So examples are important. Fine, bache, examples are important. Done. So now, bache, as I said, that we will see some modifications in the cells. What will we do? We will see some modification in the cells. Okay, we will see some modifications in the cell. Let me explain it. Let's say if this cuboidal cell, if it will be having finger-like, right? If it contains this finger-like processes on one side, if it contains these finger-like processes on one side, so these are what? These are the microvilli, isn't it? These are the microvilli, right? And these microvilli, bache, they are meant for absorption. Okay, they increases the surface area. These microvilli are what? They are finger-like processes. What are they, bache? They are finger-like processes. They increases the surface area, right? It increases the surface area. Yes or no? It increases surface area, and it is meant for what? It is meant for the absorption. Right? It is meant for the absorption. So these are the microvilli, right? So when these cuboidal cell, when they contain these microvilli, such type of epithelium is brush bordered epithelium, right? What is the word for such cells? It's brush bordered. What is it? It is brush bordered. Clear, bache? Clear? It is brush bordered, right? So brush bordered cuboidal epithelium, brush bordered cuboidal epithelium. So when do we say brush bordered? When it is having the microvilli. So what are microvilli? These are finger-like processes. What are microvilli? These are finger-like processes. They are meant for the absorption. So obviously they are non-motile, they are non-contractile, they do not have that 9+2 arrangement, right? It's just that they are meant for the absorption. Clear, bache? So brush-bordered cuboidal epithelium is present in your PCT. Do you know PCT? It is the proximal convoluted tubule. Right? It is the proximal convoluted tubule of nephron. Just I was showing you the nephron structure now. The cup-like Woman's capsule is there, right? So then you know that these convolutions, these foldings are present, isn't it? These convolutions, these foldings are present, isn't it? Isn't it? And then what do you have here? See this? There will be the loop of Henle. Okay, so here in this PCT, in this proximal convoluted tubule, you will see the brush bordered cuboidal epithelium. Why? Because 70 to 90% of absorption will be done here. Okay, 70 to 90% of the absorption will be done here. Clear, bache? So PCT is the example, right? PCT is the example. So in the case of nephron, here you will find brush bordered cuboidal epithelium. Brush bordered cuboidal epithelium. Clear, bache? And this part here, this part here, what is it? It is the collecting duct, right? It is the collecting duct, isn't it? So in the collecting duct also you will see the ciliated. Okay, here you will see ciliated cuboidal epithelium. Now what is the meaning of ciliated cuboidal epithelium, bache? What is the meaning of ciliated cuboidal epithelium? That these cube-like cells, okay, these cube-like cells are having small hair-like structure. Okay, so these hair-like structure, they are meant for the movement, of course. So it is cilia, right? Cilia. Clear, bache? What is it? It is cilia. Done. So cilia, obviously, right? Hope you remember this. When you talk about cilia, you remember 9+2 arrangement of microtubules is there. 9+2 arrangement of microtubule is present here in cilia, right? Do you remember this, bache? So on the free end of cuboidal cell, you will find cilia. Okay, so you will see it in the collecting duct. So cilia is cylindrical, it is motile, it is contractile, help in the movement, right? So it will help in the movement in a specific direction. Fine, bache. So cuboidal ciliated epithelium is present in the collecting duct. Fine, it is present in the collecting duct. Done? Sure? Okay, so next is your columnar epithelium. Next is your simple columnar epithelium. Simple columnar epithelium. Now what is the word here? Columnar cells are there. Bache, they are tall and slender and the nucleus is present here near to the base. Okay, so these cells, they are tall and slender and nucleus is present near to base, isn't it? It is present near to base. Clear? Tall and cylinder, nucleus is present near to base. Clear? Clear, bache? So it is mainly, right, columnar epithelium. These cells are mainly help in absorption and the secretion. How will they help? They help in absorption and secretion. So in gallbladder, in liver, you will see columnar epithelium, right? So when you talk about the examples, bache, you will see the gallbladder, you will see the liver. Okay, gallbladder, liver, and here also you will see various modifications. Just allow me a minute, okay? Done. So simple columnar epithelium, it is tall and cylinder. It is tall and cylinder. Nucleus is present near to the base, meant for absorption and secretion. Examples may gallbladder and liver is present. Right, bache? So here also you will see the modifications. Okay, you are going to see various modifications. You are going to see various modifications here. Like again, see columnar cells. Okay, they can also be brush bordered. So bache, if they are brush bordered, what is the meaning of being brush bordered? What is the meaning of being brush bordered? That they are having the microvilli. One free end is containing microvilli. So these columnar cells, they can also be ciliated. Okay, ciliated columnar epithelium. That is, cilia is present. Fine, cilia is present. That hair-like structures are present, which will help in the movement. We will see it in fallopian tube, right? Examples are important. I'm repeating it again. So these columnar cells, they can be glandular. What is the meaning of being glandular? Glandular means secretory. It is going to secrete something, right? So whenever we use the word glandular, glands, they used to secrete something. Right, they used to secrete something. And even here in columnar epithelium, you will also see brush bordered glandular epithelium. Brush bordered glandular epithelium. Okay, even you are going to see that. Fine. So when it is brush bordered, right, simply brush bordered, microvilli are present. So such type of structure you will see in the case of gallbladder. Okay, gallbladder is the best example here. So if microvilli are present, means it is meant for the absorption. Okay, it is meant for the absorption. Clear, bache? It is meant for the absorption. Done? Done. So if it is glandular, brush bordered columnar epithelium, so you will see it in the, if it is brush bordered, see, it is brush bordered, means microvilli are there, plus it is secretory. Both the functions are there. It is having microvilli and it is secretory. Such columnar cells you will see in small intestine. Okay, you will see it in small intestine. You will see it in your duodenum, ileum. Clear? Clear? Done? So you will see it in duodenum, you will see it in ileum. Done, bache? So ciliated columnar, when cilia is there, which will help in the movement. Okay, when cilia is there, which is help in the movement. So it will be there in the fallopian tube. So in fallopian tube, for the movement of ovum, we need it. So it is important MCQ. Clear, bache? It is important MCQ. Done. Okay, done. Fine. So brush bordered gallbladder, ciliated में fallopian tube, and then, and yes, even in the brain we have the ventricles. Okay, even in the brain we have the ventricles, we have the cavities, so there also the ciliated epithelium is present for the movement of the flow. Clear bachche? For the movement of the flow. Clear bachche? Sure? Sure. Huh? Is it clear? And yes, when it comes to the glandular, it is present in the uterus. Okay, it is present in the stomach. So different examples are there. So that is how these modifications are helpful. So whenever the word is brush bordered, always remember we are talking about the microvilli. Okay, whenever the word is brush bordered, we are talking about what? We are talking about the microvilli. Clear bachche? Clear? So ciliated fallopian tube, important MCQ, brush bordered glandular in the small intestine, simple glandular in the uterus, stomach, colon, rectum. You will find it there. And yes. Right? So this is what you need to remember. And yes, one more modification is there in the case of columnar cells. They have the stereocilia. Okay, in the columnar cells, you will see that stereocilia is also there. You know, stereocilia. So stereocilia, they are basically, you know, conical projections. What are they? They are conical projections. They also increase surface, it increases surface area. Fine, they increases what? They increases surface area. Okay, they are also non-motile and non-contractile. They will not even contract, so obviously not helpful in locomotion, even non-motile. Okay, bache? So stereocilia. So columnar cells having stereocilia, it is present in your epididymis, and vas deferens. Fine, in epididymis and vas deferens. Clear bache? Epididymis and vas deferens. Clear? Sure? Sure bache? Stereocilia. Now is pseudo-stratified. Next is pseudo-stratified epithelium. Bache, do you know where this true pseudo-stratified epithelium is present? Yes. Do you know where is the pseudo-stratified epithelium is present? Anyone in the class? Any idea about it? Yes. Pseudo-stratified. Priyanka? It's easy. But you see, pseudo means false. False. Stratified means multi-layered. Pseudo means false. Stratified means multi-layered. Pseudo means false. Stratified means okay, multi-layered. So here actually the epithelium is single layered, right? But the arrangement of cells, like some are smaller, some are taller, right? Some are smaller, some are taller. So the arrangement is in this way. Right, so in pseudo-stratified, actually the layer, right, it is single layered, but it looks like it is single layered, but it looks like multi-layered. Are you getting it? It looks like multi-layered. So that is the meaning of being pseudo-stratified. So ultimately, it's the example of simple epithelium, but it looks like the multi-layered pseudo-stratified epithelium. So cells are again columnar types. You know, these are basically the modification of columnar epithelium. Fine, bache? Modification of columnar epithelium. Clear? Clear? Done? Let me write it here. So these cells are the modification. What are they? They are the modification of columnar cells. You will see in this epithelium that nucleus are present at different places, right? Or different positions. Like if you will notice here, some cells are large, nucleus is here. Some cases nucleus is here. So nucleus position is not same, right? But all the cells right? All the cells, they are lying on the same basement membrane. Okay? All the cells, they are lying on the same basement membrane. So it looks like multi-layered, but it is not multi-layered. It lies on the same basement membrane. So these cells are the modification of the columnar epithelium, right, bache? So it is pseudo-stratified, looks like multi-layered, but actually it is not multi-layered. So the best example is your, right, the best example is trachea, bronchioles, okay? It's a previous year question, bache, trachea, bronchioles. So basically in our respiratory epithelium you will see this. In nasal chamber also you will see this. Okay. So in the trachea, in the bronchi, in the bronchioles you will see pseudo-stratified glandular columnar epithelium. It is pseudo-stratified glandular columnar epithelium. It is also glandular there. Okay, it is also glandular there. Is that clear? Yes, bache. Is it clear? It is also glandular there. Fine. So this is pseudo-stratified. Examples are important. If there is any doubt, please tell me. Please tell me if there is any doubt. So see the examples: cuboidal, PCT, right? Nephrons are having microvilli, columnar epithelium, metol and cylinder cells are there. So pseudo-stratified glandular columnar epithelium is also there. So I will share the PDF with you all. Fine. Fine. So ciliated epithelium can be cuboidal, it can be columnar. So it is for your bache quick revision. Fine. It is for your quick revision. So I will share these notes in the Telegram group. Fine. With the position, everything is mentioned as per the NCERT. Fine, bache. So whenever we use the word glandular epithelium, na, means it is secretory. Okay, means it is secretory. So first of all, let's discuss the compound epithelium and then, okay, first of all, let's finish the compound epithelium and then we will talk about the glandular epithelium. But yes, before that, let me show you the diagrams here, bache. So I was telling you about the cell junctions, na, that in epithelial cells, no intercellular matrix is there. So we need to hold the cells together, right? So here you guys can see the cell junctions. So there is a little intercellular material present between cells. There are specialized junctions present between the cells of the epithelium that link the individual cells. Clear? Okay. So tight junctions, they stop leakage. Adherence junction, cementing neighbor cells. Gap junction facilitates the cell communication. So have a look of these diagrams. Fine. So see the gap junctions. So gap junctions may, as I said, which protein will be there? Your connexin. Which protein will be there? Your connexin. Okay. So adhesion, right? Joining. So see the disc shaped protein, your desmosomes, and here also proteins are present, filaments are present, and here you will see these are the intermediate filaments which are there in the cytoplasm of the cell. Okay, the tight junctions. Have a look. Fine. So I hope it is clear. I hope it is clear. Better start revising then, Soumyadeep. Done? See the cell junctions. Okay. The cell junctions. Fine. Look at the diagrams. Tight junctions, adherence junctions, your desmosomes basically, your gap junctions. Right, your gap junctions. Done, bache. So now we need to start the compound epithelium. Okay, so in the compound epithelium, as I said, it is multi-serrated. Multi-serrated means it is multi-layered. Fine. Multi-serrated means multi-layered. So it is not made up of only one layer. There will be so many layers. Okay, there will be so many layers. Guys, you can join my personal Telegram channel as well. You can join our official channel as well. Right, I will share the PDF there. And now please have a look. So what are we discussing? We are discussing multi-serrated epithelium. Okay, multi-serrated epithelium. Many layers will be there. So see, layers will be so many. Okay, let's discuss it in this way. What are we discussing? We are talking about the compound epithelium. What are we discussing? We are talking about the compound epithelium. So I told you that here you will have stratified epithelium, right? In stratified, you will be having stratified squamous epithelium. Stratified squamous epithelium. We are having stratified cuboidal epithelium. We have stratified columnar epithelium. Okay, stratified columnar epithelium. Done. And the another one is transitional epithelium, also known as, it is the transitional epithelium, which is also known as urothelium. What is it? It is also known as urothelium. Clear, bache? It is also known as urothelium. So this is what we need to discuss. Now see, it's very simple, bache. Stratified squamous. One word is clear that it is going to be multi-layered. Okay, one word is clear that it is going to be multi-layered. So obviously there will be the basement membrane on which all the cells are placed. So here you can see there will be many layers, like let's say 3 to 4 layers are there, 5 to 6 layers are there. But when we are using word stratified squamous, right? When we are using the word stratified squamous, so the topmost layer will always be having, because it is squamous. So topmost layer is of flat cells. It is of that squamous cells. Are you getting it? It is of that flat cells. It is of that squamous cells. So if it is stratified cuboidal, so topmost layer is cube-like. Topmost layer is, right, of cuboidal cell. And when it is stratified columnar, topmost layer is having the columnar cells. Right? So rest layers can have different types of cells. Here you can see, right, you can see, you know, cube-like cells also, columnar cells also. But when it is stratified squamous, topmost layer will have flat cells. Topmost layer will have cube-like cells. Topmost layer will have columnar cells. Fine? Fine? And now this is the transitional epithelium, urothelium. It is bache stretchable epithelium. Do you know that? It is the stretchable epithelium. Yes, it will get stretched. Okay, it will get stretched. It is stretchable epithelium. Fine, fine. And here, this stratified, this one is non-stretchable. Let me explain it to you first. Fine, it is non-stretchable. So let's start with the, okay, let's start with the transitional epithelium first. That is your urothelium first, which is the stretchable one. Okay. Done. So firstly, let's start with the urothelium. But urothelium and the transitional epithelium, it is going to line the urinary bladder. It is going to line the ureter. I hope you remember the structure. Let me draw here. So you know, in our excretory system, we have the pair of kidneys and these two, this pair of tubes, right? This pair of tubes, what is it? This pair of tubes, what is it? It is the ureter, and here this conical structure is what? It is the urinary bladder. Here the urethra and opening is there, right? So when you talk about this urinary bladder, okay, when we discuss about this urinary bladder, see this urinary bladder. So here in this urinary bladder, you will see it is lined with this transitional epithelium. Even some part of this ureter is also lined with this transitional epithelium. So when you talk about this transitional epithelium or the urothelium, right? So it contains many layers, obviously. So if you talk about the, like, see, first of all, here we will have basement membrane. Here we will have basement membrane. What are we going to get here? We are going to get the basement membrane here. Okay? So here, just above the basement membrane, 2 to 3 layers of some globular cells, Okay, so here what will you see? Right, some not globular, some cube-like cells. Okay, so you will see some cube-like cells. Fine, and then you will see here some, now the shapes are obviously not so relevant, but just to explain it to you. Okay, so here you will see what? You will see 3 to 4 layers of some, polyhedral cells. Polyhedral cells. Fine. And then the topmost layers here. The topmost layers here. Again, they will be, kind of globular. They are oval. Fine. Oval in shape. Fine. They are oval in shape. So when you talk about this urothelium, also known as transitional epithelium, so you will see that outermost cells are oval in shape, innermost are cube-like, in between some polyhedral cells are present. Now what is the point here, bache? See, you know that we can hold our urine for some time, isn't it? We can hold our urine for some time. Do you know that? We can hold our urine for some time. Like let's say you are attending a class, right? And there is an urge to urinate. So you can control it for some time. But after that, right, you can just only control it for some time, but not later than that. But not after that. Hai na? After that, it will be very difficult for you to hold your urine. Why is it so? Why? Like if let's say if I am taking your lecture, right, and if I need to urinate, I can hold it for some time. Only up to a limit I can hold it, but not after that. How is it possible, right? So it is possible because of this transitional epithelium. Now you must be thinking, how ma'am this transitional epithelium can do it? See, as I said, topmost layers are having some oval cells. Now here in this epithelium, right, it is stretchable epithelium. What type of epithelium it is? It is stretchable. So when the volume of urine will increase here, these oval cells, they will become flat. They will become distended. The distension of these cells will occur, right? So with the cell, it is oval now. The cell, it is oval now. It will become flat like this. So obviously it will accommodate more urine within. Are you getting it? It will accommodate more urine within. So what is going to happen when the volume of urine increase here, right? These oval cells, they will become, yes, bache, they will become stretchable. They will become flat. Okay. Okay. They will become flat. Are you getting it? Yes or no? Are you getting it? Yes. Are you getting it? Done. So initially it was oval, now it is becoming flat. So obviously the volume of that urinary bladder will increase. So yes, we can hold the urine for some time. But after that, no, there will be no further distension. The cells will not become more flat. So obviously in that case, they will pass out the urine now, right? It will be important for us to pass out the urine, isn't it? So that, shukriya. So that is the most important point. Okay. Okay. So that is why it is the stretchable epithelium. Fine, it is the stretchable epithelium. Done? Okay, clear, bache? Yes, is it clear? Sure, sure. So that is why it can accommodate that urine. So basically when the urinary bladder is half filled or little more than half filled, we get that urge to urinate, but we can hold it for some time. So that is the beauty of this transitional epithelium. And here in between these cells, you will see interdigitation. What is the meaning of this interdigitations? You will see that one cell, right, the plasma membrane is forming finger-like projections, right? It is present in the cytoplasm of another. So structure like this. So that is how these cell layers are connected. Fine, that is how these cell layers are connected. So this is your transitional epithelium. This is your urothelium. Is that clear, bache? Yes. Is it clear? Okay. Okay. Sure. Sure. So next is your stratified epithelium, in which we will talk about the stratified means multi-layered. Okay, stratified means multi-layered. So stratified squamous epithelium. Okay, stratified squamous epithelium. Now, bache, it is multi-layered. So one thing is again common, that here you will be having the basement membrane, because the cells are compactly arranged, so they do not have the intercellular spaces, they do not have the intercellular matrix. Okay, so near to this basement membrane, here you will see some, you know, cube like cells. Do you know that? Here you guys are going to see some cube like cells. Fine. What will you see here? You are going to see some cube like cells. These are the cuboidal cells. Fine. Innermost layer is cuboidal. Innermost layer is what? Yes, bacho, it is cuboidal. Now this layer is mitotically very active. Do you know that? It is mitotically very active. Means it will undergo mitosis very quickly. Okay? It will undergo mitosis very quickly. So it will add more and more and more cells here. Fine? It will add more and more and more cells here. Okay? So this layer, because it will divide, it will add more cells here. So mitotically it is very active. So we call it as stratum germinativum. Okay, what is it? It is stratum germinativum. Okay, so this is the one which will add the cells here. This layer is stratum germinativum. Clear? Stratum germinativum. So here you will see some, you know, polygonal cells here. Many layers of, I know the shape is not proper, but you please try to understand. Okay, so polygonal cells are there. Many layers of polygonal cells are present here. And the most important is here, because it is stratified squamous, so the topmost layer is made up of flat cells. Clear, bache? The topmost layer will be having that flat cells. That is why you are saying stratified squamous. So in stratified squamous, you have further two categories. One is keratinized, stratified squamous epithelium. One is keratinized, stratified squamous epithelium. Another is non-keratinized, stratified squamous epithelium. Non-keratinized, stratified squamous epithelium. Is that clear? Yes. So, bache, keratinized, keratin protein. Okay, what is the word here? It is keratinized. Here we are talking about the keratin protein. What are we discussing? We are talking about the keratin protein. Are you getting it? Are you getting it? Right? So what happened? Like even in the case of our skin, right? Our topmost layer is kind of dead layer, right? It is having the keratin in it. Keratin is a protein. Now what happened? Right? The topmost layer, it will replace the cytoplasm, and in the place of cytoplasm, we will get the keratin protein, right? Right? That is why it is also known as horny layer. Fine. It is also known as cornified or horny layer basically, right? Now, because of this keratin deposition, the water will not get lost from our body. Okay. Okay, so it is keratinized, stratified if keratin is protein in that cells. Fine, right? So topmost layer is the dead layer. Why? Because topmost layer is having, in the cells we are having the keratin. So now that cell is not living, that cell is a dead cell. So everything will be replaced by the keratin protein. Clear, bache? Everything will be replaced by the keratin protein. Fine, so I'm writing it again. If keratin is present in, if keratin, clear, bache? If keratin is present in topmost layer, layer become enucleated. It is actually the dead layer. So yes, in the case of skin, you will see this. Fine. So you will see it in the case of your skin. Fine. In the case of your skin. So skin is the example. Our nails, you know, na, nails. Keratin protein is there in the nails. Here, they are the examples. Clear, bache? They are the examples. Done? Done? So if keratin protein is not there, outermost layer is living. So here you have the example of cornea of eye. cornea of eye. So cornea of eye is multi-layered and it is not having the keratin. So the topmost layer is living, it is not dead. And buccal cavity, esophagus. So they all are non-keratinized stratified squamous. Fine, non-keratinized stratified squamous. Done, bache? Yes, is it clear? Sure? Is it clear? So next is, bache, after this, you have stratified cuboidal epithelium and you have stratified columnar epithelium, right? Stratified cuboidal, stratified columnar. Now, do I need to explain it, bache? You know, na, it will be multi-layered. The topmost layer will be cube-like. The topmost layer will be columnar. The topmost layer will be cube-like. The topmost layer will be columnar. So here, right? So this stratified cuboidal, it is present in your sebaceous glands. It is present in your mammary glands. In mammary glands are also the modified sweat glands. No. So it is also present in sweat glands. Fine. It is also present in sweat glands. So sweat glands are having the stratified, right? It is having this stratified, basically stratified cuboidal is present here in this sweat glands, in the mammary glands, in the sebaceous glands. Clear, bache? Clear, bache? And here you will see, find it in the case of pancreas. You will find it in the case of epiglottis. That's all. That's all. So in the secretory duct, of sebaceous gland, of mammary gland and sweat gland, stratified cuboidal epithelium is there. So that's it. That's all. Rest, right. That's it. That's all. So see, examples are given. Fine. So junctions of rectum and anus are also having this stratified columnar epithelium. So you guys can see the diagrams here. So diagrams are also showing the glands. Okay. So unicellular, single-celled glands are there, like your goblet cells, mucus, right? Mucus secreting cell, goblet cells. They are basically secretory, glandular. They are providing us some secretion. So look at this diagram, the multi-layered diagram. See, the topmost layer is made up of irregular cells. So obviously it is stratified squamous epithelium, right? It is stratified squamous epithelium. Done, bache? So this is what we are going to cover today, right? Rest we will discuss in part 2. So you know what you have to do. If you like the session, please put it in the comment section. Let me repeat it again. I will take another session on plant kingdom. So your part 2 will be discussed then. Okay, don't worry about that. But as of now, whatever chapters we have covered so far, please revise it. Try to make the notes. It is of no use if you will be, if you will start studying in the last 3 months or 4 months. If you will start now, it will be easy for you. Fine, it will be easy for you. So start now. So, bache, I will share the PDF in the Telegram group. Okay. So tomorrow is the last day to join the NEET Energized batch. Be the part of that batch. That batch is amazing. The team is amazing. We are taking the tests from students. Doubt sessions are there. Right. The in offline coaching, you're going to provide 1 to 2 lakh for such content. And now you just need to pay 15,000 rupees. Okay, so test, revision, practice, recorded classes, you are going to get everything. Fine, you're going to get everything. Fine. So start revising properly, and tomorrow I will post one strategy video, how to score 360 out of 360 in biology. Do check that. Thank you so much, Suhashini. Okay, do check that. Fine. So take care. Bye-bye. See you all. Thank you so much for watching, and now you know what you have to do. Please put a comment. Okay, any suggestion, any doubt, or you like the session or not, please put it in the comment section.